Hi, everybody. Welcome back. We are in the breakout session for GRI animal models and gene therapy. We're going to hear from two speakers today. Oops, there went an earring. We're going to hear from two speakers today, and our first speaker is Dr. Amy Ramsey. Dr. Ramsey, go ahead, take it away.
Good morning. Thank you so much. I'll just pull up my my PowerPoint. Sorry about that. All right. Okay, here we go. Thank you. So I'm going to be talking about two different types of gene therapy that we're working on in our lab at the University of Toronto. Um, let's see. So, when I think about gene therapy, there's really three big uh, methods that we have in our toolbox, although there are a lot of variations on this theme and there are new ones being developed as well. So this is certainly not comprehensive. But so the three big ones that I think about are gene replacement. And really, that idea of replacement is that this is for a person who has a completely non-functional gene or genes, and we're going to replace that with outside DNA that we're bringing in that's going to fill that gap that the person has. Um, so essentially, it's delivering DNA to replace something that's missing. ASO stands for antisense oligonucleotide. And what that means is that the sequence of the nucleotides is the exact opposite of what the mRNA is that is making the protein. And so when you add in those nucleotides, they stick to the RNA and they can either increase the amount of protein that's made from that RNA or decrease the amount of protein that's made from that RNA. And then the third one is to use CRISPR-based approaches. And CRISPR is really just a protein that, can drag along other enzymes to a specific target area of the genome. And so the CRISPR part is what brings it to the exact correct location. And then you can drag along with it other enzymes that can make the modification that you want. So I'm going to talk about gene replacement for the most of it, and then I'm just going to touch on a CRISPR-based approach that we're just starting on. So in the case of the GRIN patients and also the GRIA, GRIC, and GRID patients, often this is a single nucleotide change, and it's only on one of the two genes that we inherit from our parents. And so, because of this, it's not really that people have absolutely missing, gene function. It's just that it's one of the two copies is not working properly. And so, it seems strange that you would think about replacement as a strategy. But what we thought is that the way all of these channels are assembled is actually that you need four subunits to come together. And really, if even one of those is problematic, then it's a problem for the whole receptor. So here in this picture, what I'm showing you is that the normal way that a receptor is built is that you have two of one subunit type and two of another subunit type. And in my case, we're studying GRIN1. So let's imagine that we have, in the normal situation, two functional GRIN1 copies and two functional GRIN2 copies. In the patients, we have a pool of some of them are functional and some of them are not functional. And so when those all get assembled, it's really random what gets put together in a receptor. So you have some of the receptors are functioning normally, but if you have even one non-functional subunit, the whole receptor is not working properly. And if you have two, it's certainly not working properly. So what we thought is, well, let's just shift the odds so that when receptors are assembled, it's more likely that you will have assembled ones where they have the two functional subunits. So that was our goal here. All right. And the way that we thought first to add in that extra copy was through adeno-associated virus. The reason for that is that, this is already being used as a method for gene replacement in other neurological disorders. So that means that it's already gone through a lot of toxicology and a lot of regulatory review. And so it's, let's just start with something that has already passed those really big hurdles. And so, this was our strategy. And we did this really with the help of homology medicines. And that partnership was really built on CureGrin bringing us together with a company that specialized in the AAV part. So they knew about AAVs, we knew about GRIN, and together we could make this AAV. So I'm just giving you an example of some of the studies that we've done. One, we had to do a lot of dose finding. So we started with our medicines and we would inject them into the brain in different doses to find out where we could get therapeutic effect without causing problems to the brain. Because, if you put a virus in the brain, even something as innocuous as this AAV that doesn't replicate and doesn't make new virus, the brain will still have an immune response to it. So what we're looking for is what's the dose that you can give where you don't get a lot of neuroinflammation. And so here, what we're looking at is 10 to the 10 virus, 10 to the 9, 10 to the 8, and 10 to the 7, delivered right into the brain. And then we're looking for markers of inflammation. So in the purple, you can see that 10 to the 10 creates a lot of microglial activation, these purple specks, and it creates a lot of astrocytic activation. So this is indications that neuroinflammation is happening. And so when we get down lower, you have less. And we really thought that 10 to the 8th was a nice dose because we didn't see any indications of neuroinflammation. And what we actually decided to do for our next studies was to choose an intermediate dose. So we have 5 times 10 to the 8th. So this is kind of a low dose, according to what we had been kind of advised by homology medicines to start with for our kind of intrabrain administration. And so we wanted to see if this dose could be effective. And also we needed to test this idea that if you deliver more wild type GRIN1 cDNA, does this actually benefit or overcome the problems that the variant has? So here we're using mice that carry a patient variant. And the first one that I'm going to show you is the results that we have with the variant GRIN1 Q536R. And this is one that has a variant in the glycine binding domain. So these don't have any problems getting to the surface of the neurons. And really their main problem is that they aren't activated by glycine the way a normal NMDA receptor should be activated. So excitingly, we saw amazing improvements here. So what did we do? We injected this virus locally into this structure in the brain called the hippocampus on both sides. Then this was done in Graham Collingridge's lab by Dr. Patrick Tidball, and really amazing electrophysiology where he stimulated axons and then listened to the collection of neurons that are activated by those axons. And here's what we see. He did a lot of different studies to measure axonal integrity and how much glutamate was being released, how many AMPA receptors were being activated. So really, I'm just showing you the part that's relevant for NMDA. Everything else was normal between the Q536R animals and their wild type litter mates. So here's what we're showing you is that when you stimulate with different intensities, you get more and more of a response. In the heterozygous animals, the Q536Rs, there's a diminished response with this electrical activation. And with the application of this AAV, we actually see this restored back to wild type levels. So this is one example trace. And this is what we see when we studied 6 different animals. So these are wild types treated with just the buffer, or the heterozygous animals just treated with a buffer. And then these in yellow are the animals that are treated with the AAV. And when you sum that all together, you can see that the NMDA receptor response is less in the heterozygotes, but when you give the AAV, it's the same as wild type, which is just phenomenal. It's beyond what we could have expected. So I don't know how to convey this to you, but this was really exciting for us. It really showed us that, okay, this strategy can work. The other thing that they can do is to look at experience-dependent plasticity, or when you activate a synaptic connection over time, it should strengthen. And that process relies on NMDA receptor function. So this is another way of looking at NMDA receptor function. So here they are activating these axons, stimulating these axons in a burst pattern, and then recording and seeing how these neurons over here respond to each electrical kind of pulse after they've been highly activated. And so what you can see is this is before that burst, and then after you get a much bigger response in the wild types. In the heterozygous animals, that's diminished. So this would be the patient variant animals. But with the AAV, that plasticity is restored. So this is saying that these NMDA receptors, not only are they, responding normally to stimulation, but they're also allowing this process of learning at the synaptic level. Okay, so this is one animal that has a change in the ligand binding domain, but we actually have 2 animals that we study in the Ramsey lab. And so we did the same thing. Oh, I forgot about this. Yeah, we did the same thing in another variant. But I just wanted to say that this effort was the dream team. So Peter Finney in my lab said, I'm working with such great scientists. I feel like we've got the dream team here. So the dream team is Phoebe Yan, who is a graduate student, Peter Finney, who is a research associate in my lab, and Patrick Tidball, who's a postdoctoral fellow in Graham Collingridge's lab. So the 3 of them did this. Phoebe and Peter were doing the AAV part of it, and Patrick doing the electrophysiology part of it. Okay, so this is the next step that the dream team did. They did it with another animal. So this one has a more deleterious variant. It's Y647S. And so this one is really at a very important part of the channel that affects the flow of ions. It's right at that ion gating part of the channel. And so here you can see the wild type responses. The heterozygotes have really no synaptic NMDA receptor function. And then when we give the AAV treatment, it's markedly improved. So here we only have 3 animals. I think that since then the team has done another 2, and we're waiting on the results of the last animal, but the results from the 2 that I don't have the data for are consistent with this. So we're just getting very similar results from experiment to experiment, which is just great. So here what you can see is that this NMDA receptor current in the synapse is really lost and is greatly improved. And here is LTP, which is that learning component that's mediated by NMDA receptors, and you can see that it is diminished in the heterozygous animals, and it's back to wild type in the AAV-treated animals. So this is my big, exciting story to tell you. But we really ultimately want to think about a medicine to treat people, and intracranial is not the best way to do it. So we'd ideally like to be able to make a medicine that we could give IV, that could go into the brain and achieve our goals. And that's what they have for SMA, which is currently treated with a gene replacement strategy, and also Duchenne's muscular dystrophy, which is another neurological disorder that is treated with a gene replacement strategy. So we know that IV administration can work for neurological disorders, and we want to do that for the GRIN patients as well, and GRIA and GRIC eventually. Not my lab. We can't take on everything, but this is where we're going next. So we've been doing dose finding studies, and so here in the bottom, we're looking for the expression of our cargo, which is the GRIN1 that's been tagged with a special tag so we can track it and keep it separate from what's normally expressed. And so you can see that with increasing doses, you can see more of this purple, which is the evidence of the GRIN1. Now, this looks really faint, but I just want to tell you that when you look for NMDA receptors, you should not be looking for blasting signals, because these are things that are, expressed at really low levels, but, kind of evenly throughout the brain. So really, this is enough, we believe, to achieve a functional rescue. So here, this is looking at the amount of cargo that's being expressed, looking at 3 different doses in the brain, in the heart, and in the liver. So there's a lot that gets into the liver, there's a lot that gets into the heart, and there's a lot less that gets into the brain. The reason why we care about this is that kind of the effects that are seen with AAV that we know about from treating SMA and treating DMD are liver toxicity and cardiotoxicity. So we really want to note what's going on with the liver and the heart, and part of that is knowing how much virus goes there, and we're going to be looking really carefully at those organs to make sure that they are okay. So that's just a little snapshot of some of the things that we're doing. We have some preliminary results looking at the weight. And so here we've given these animals IV, the AAV, so intravenously, and we were measuring their weight every week. And really, I think the take-home message is that, we have very low N. There's not a lot that we can say. They don't seem to be getting worse. They're not getting sick, really, especially at these low doses. That their weight, they actually seem to be gaining weight nicely, and there might even be some benefit, but really too early to say at this point. We've also done some really simple behavioral tests. The main goal, again, was just to look at expression. So these are really low numbers of animals, and I don't want you to take too much away from this, other than just to say we're doing this. And so one of the things we look at is where the animal prefers to be when you give them a choice between light and dark. And usually a wild type mouse will prefer to be in the dark. It's safer, but the mutant animals prefer to be in the light. I think it's because they have some visual problems, or maybe some anxiety. There could be many reasons why they do that. But we did see in these treated animals a pattern that was more similar to wild type. And again, so one of the things we often do with these animals is we look at their behavior when we put them in a new environment. So we're just recording how much they move around. And that novelty will often cause them to run around more or less than the typical animal. So it's a really nice screening test. But you can see that these particular mutant animals are not that different from wild type. And so we can't really use this locomotor activity with this particular variant mouse. However, we did notice that their repetitive movements were changed a little bit. So we're looking at this as perhaps there's something there that we can tease out as a readout of whether this virus is working or not. Okay, so those are my little snapshots of what we're doing with the gene replacement. So what I'll say is that there's good news and bad news. The good news, gene augmentation seems to be working. I'm calling it augmentation rather than replacement because it's really more that we're adding extra in, not so much that we're replacing something that's missing. Okay, so this is great. We've filed a provisional patent, which is what has enabled me to talk about this with you freely. And I think it's, it's been so hard for us to keep quiet about this, but we wanted to make sure that, eventually we'll need some company to help us actually bring this to the clinic. And we didn't want to lose out on that opportunity if we didn't have it protected. So we made sure that it was protected. And then now we can really start to share this information with you. So I'm really happy that we can give some positive news in that progress in this field. And we have a great team that we're working with. So not just the Collingridge and Ramsey labs working together, but two other labs at U of T that are doing CRISPR-based research and also lipid nanoparticle research. And I'm going to tell you about that in just a second. Okay, but there's some bad news that I need to temper, the enthusiasm a little bit. So when we did these studies injecting into the hippocampus, we haven't so far seen any behavioral benefits from that. So we're starting to see some limited benefits with the in vivo in the intravenous administration. But I want to say that right now we don't have really strong, slam dunk evidence of that. So I think that there's still really a lot that needs to be done before we can say, okay, we've got something here. The other thing that's really been very frustrating, sad, I'm just really disappointed about this, is that just the problem that happens with all biotechs is that when, the economy has a problem, venture capital tightens up and isn't available. And so they were acquired by Q3 Bio, which is really not focused on neurological disorders. So we're going to start experiments with a new capsid. So this is just to say that we've made progress, but we might have to step back in order to go forward. Okay, so that's that. And the other thing that I want to just share with you is this idea of using a different delivery system than adeno-associated virus. So there's something called a lipid nanoparticle. And what is that? So you start with RNA and here, and then you coat it with lipids. And the idea is that by coating it with lipids, it is the Trojan horse that covers up this RNA and allows it to fuse with cells. And that RNA gets put into the cytoplasm of the cell. And from there, it can be made into protein. So this was work that was done by Lyra Vanya, a graduate student in my lab, in collaboration with Bowen Lee's lab at University of Toronto. So Bowen is an expert on these lipid nanoparticles, and she asked, can you help us with this? So this is the first test where they tried all of these different combinations of lipids, and they were looking at whether they could deliver the cargo, which was the firefly luciferase that makes light, whether they could deliver it into astrocytes or neuron-like cells. And you could see that some of these combinations were really good. Some of these combinations were really bad. This one is the same or very similar composition to what's used for the Spikevax COVID vaccine that Pfizer made. So this is actually what was used to deliver the mRNA that we all got in with our COVID vaccines. So this is just showing that what we did after we chose, some of these LNPs, then we would, actually deliver the same light producing cargo into the brain, wait 24 hours, and then this is imaging just to see how much light is being produced. And this is going through the skull and through the hair. So you can imagine there's quite a bit of light there this camera is able to pick up. So this is showing us that sure enough, this LNP is able to deliver the gene, and it makes the functional protein that can then make the light. So this is telling us, yes, these lipid nanoparticles get into brain cells, and that their cargo gets made into what you want to make. So we have been doing this with, instead of the firefly luciferase that makes light, we've been doing it with the GRIN1 mRNA. And again, the idea is that, even if you only have one bad copy, that's problematic for 3 out of 4 of your receptors. So let's just add some more and see what happens. So we're doing that now. We're doing a lot of studies of the tissues, the brain, the heart, the liver. Then we'll be doing some electrophysiological recordings, and ultimately we'll be looking at their behavior. So I think this is just one little slide showing kind of the things that we're doing to see if the GRIN1 mRNA is kind of being made into protein. And so this is an mRNA that's tagged with GFP, and we can say, sure enough, it is being delivered, and not just the GFP tag, but the GRIN1 itself is being delivered. And these were done in animals that have very low levels of GRIN1, so we can see what's being delivered above the wild type levels. Okay, so that's all I really want to say about the LNPs. It's just to say that, the advantage of the LNP is that they don't have the same size limitation for the cargo. So one of the things you may have heard about AAV is that, it really can only carry things that are about 5 kb. And there's a lot of things that are too big to go in. And the GRIN2 genes are just at the kind of threshold of what is possible. And also some of these CRISPR mechanisms are too big to go into one AAV. So the LNP could allow bigger cargo to go in. And that's what we're trying with the CRISPR-based editing. And I'm not sure, I might be running out of time. So really all I want to say is that we're going to try this type of editing. First, we have to do tests in cells, then we can try it in mice. And then there's a lot of stuff that we'll need to do. So this is a very preliminary stage, just to let you know that we're hoping to do this. I'm not going to go into this, what's going on in prime editing. If you need to know, I can tell you. So just to summarize, we have some patient variant mice. We're pursuing LNP and AAV. We're also going to try and do prime editing. It requires a lot of studies and where it's going in the body, how long it lasts, whether it causes immune activation, liver toxicity, and to measure efficacy, there's a lot of studies that need to be done. And with that, I'll just say thank you. I want to highlight Phoebe and Patrick, part of the Dream Team, and Lyra, who has been doing the LNP studies. And thank you so much to all of these precious ones and their families and the support of Cure Grin. I wanted to highlight, I am really enjoying my collaboration with Graham's lab, Bowen's lab, and Basil Hubbard's lab that's doing the CRISPR stuff with us. So I'll end there. Thank you for your attention.
Thank you so much, Dr. Ramsey. There's a lot of excitement in the chat in the community. So just amazing work and really great news. There were a couple questions, but we'll go ahead and turn it over to our next speaker. If we have time at the end, we can open it up for Q&A. Otherwise, we'll be sure and capture those questions for the Ask Me Anything session. So
Okay.
now we'll hand it over to Dr. Wayne Frankel.
Great. Thank you very much. Can everyone hear me and see my slide?
Yes, we can. Thank you.
Great. Well, thank you so much. Great talk, Amy. And I'm so glad you're talking about the delivery systems and working through them. Our lab has been primarily focused on the molecular side and the phenotypes in the mouse models, what seems to work and what doesn't seem to work. Today, and thank you for the organizers, Denise, Keith, Megan, Brittany, Lauren, for inviting me. So today I'm going to tell you about an approach that's been discussed for a little while, and I've not seen a lot done on it yet. It's called knockdown and replace. It's gene therapy in a mouse model of severe childhood epilepsy, very much like the model I'll discuss is very much like a green gene. It's not a green gene, although my colleague Tristan Sands is going to talk about our work on one of the Cure Green funded projects on Green 2A in a later breakout session. So this work is a collaboration between my lab and Tristan's lab at Columbia, Scott Harper, Gene Therapy Center at Nationwide Children's, and Matt Weston at Virginia Tech. So as many of you know, severe pediatric epilepsy and related epilepsy, other nerve developmental disorders that are largely difficult to treat, has significant genetic component. They're rare or ultra rare individually, collectively. They're more and more common. There's at least 85 genes now that are known to be involved, thanks much to exome and now genome sequencing in the past 10 years. And these genes are involved, the proteins that made by them are involved in many molecular mechanisms, physiological mechanisms, not just channels and receptors, signaling proteins like CDKL5, transporters, even non-neuronal genes, synaptic proteins, and other things. Now, in cases like this where effective traditional therapies, if they do exist, are few and far between, if you're a geneticist, your conclusion is, well, how about gene therapy for a genetic disease? And that's the philosophy and approach that we've taken. But the specific approach, as you've heard from Amy and many of us have discussed, depends a lot on the gene and the molecular effect of the mutation. And so genetically, when inherited, genes could be recessive or dominant or additive. At the molecular level, the mutations we discuss them as loss of function, as in GRIN1 that Amy described, where the whole gene is deleted, or the RNA isn't fully made, or there's a frame shift that deletes it, or sometimes even a small deletion or a missense can cause a loss of function. At the other end of the spectrum, there's gain of function, where the RNA and protein is made, but it's doing something bad. And this is usually, but not always, due to missense mutations. And there's also dominant negative, which are usually, again, missense mutations, sometimes truncations and indels. They're kind of like really bad loss of function, whereby the mutant copy doesn't work right on its own, and it interferes with whatever is left, for example, the other gene copy of the normal gene. And so the model I'm going to discuss today is dominant negative for dominant negative disease. And so, as you heard, loss of function, you want to replace or add more normal RNA or increase expression of the good copy. For these gain of function, dominant negative, ideally you want to eliminate the bad RNA or change the gene itself, like Amy just discussed, the editing. But even when you eliminate the bad RNA, you may also need to add more good RNA. And this is effectively what one means by knock down and replace. And again, Amy discussed AAV. It's potentially a one and done kind of therapy because the genome exists for the life of the transduced cell. So once a neuron gets this AAV, as long as the neuron is alive, the AAV produces its therapy vector. Yes, there are issues with autoimmune responses and other bad things that happen. And as Amy discussed, there's work in other delivery systems. There's also folks working on minimizing sequences in AAV that aren't really necessary that might minimize such responses. But again, we've been focused more on the molecular side and the seizure and behavioral phenotype side. So the gene I'm going to be talking about is called Dynamin-1, DNM1, which causes a uniformly severe form of this disease, this developmental and epileptic encephalopathy, or DEE. Most of the cases occur, plurality occur less than 6 months of age. The phenotypes are largely intractable, also accompanied with severe, profound intellectual disability, nonverbal, non-ambulatory, and hypotonia. And so this Dynamin-1 encodes not a big protein as a monomer, but what it does, it assembles into these ring structures that are important for pinching off the synaptic vesicle, the thing that communicates chemical signals to other neurons. It pinches it off so it can go back into the cell to recycle, load up with more neurotransmitter, and then release them into the synapse. And so that's what this Dynamin-1 does. And so the dominant negative, not only in our case, doesn't assemble into these multimers, but it also impacts the ability of any normal monomers there to assemble. So the Dynamin-1 gene has several domains. All the known human pathogenic variants occur in the GTPase domain. The GTPase is the enzyme that provides energy for that event, or in the middle domain, which is where our mutations that we're studying are, which is important for that assembly from the monomer to the multimer. Now, previously, we published work on a DNM1 mutation called FITFL that we found some years ago at Jackson Lab when I was there. We used a selective RNA interference, or RNAi approach, delivered by AAV, and we showed it was effective against the most severe symptoms. So untreated mice have significant growth delay, they're not strong, they wobble with ataxia, and they have severe lethal seizures by the 3rd week of age in mouse. There's also some cellular pathology. With a single dose, we were able to eliminate most of these severe symptoms. The mice have very mild features afterwards. So this was encouraging, but the FITFL mutation is a little bit odd for a couple of reasons I don't have time to go into. Direct relevance to human cases wasn't completely clear. And it's true that the way we could target the mutation wasn't selective to the mutation, but to the isoform that uniquely expressed it. And that's not always the case in DNM1. So only some of the cases would have benefited from this approach. So we needed a different approach. And that's why when we thought of knock down and replace, so the concept is to knock down all endogenous DNM1 levels, locking down those RNAs, both the normal copy and the bad copy, and then replace it with a new normal wild-type cDNA that's resistant to the knockdown part of the vector, and is furthermore was engineered so it should be optimally translated as an RNA. And so you can see here this vector that was designed by Scott Harper in his lab is a single AAV that contains both genes. They're expressed each off of its own fairly ubiquitous promoter. So the microRNA is a shuttle that delivers the RNAi that knocks down DNM1. And the replacement is a full-length mouse codon-optimized, knockdown-resistant cDNA that goes off of the synapsin-1 promoter, which is expressed in all neurons. And this just shows Scott's lab pre-screening of this vector in vitro against a normal wild-type cDNA and it knocks down the amount significantly, whereas it doesn't really touch the RNAi resistant cDNA that we have in here. So that's the approach we're taking. And I also want to add that, a benefit of this for a gene that has multiple missense variants that cause this kind of disease is this approach is in principle agnostic to the type of variant. If it works, it means that you're knocking down all the RNA and replacing it with all normal RNA. And so one drug, if you will, could potentially be effective in any of those mutations for that gene. I'm not promising that. I'm just saying that's the concept. So we made a new mouse model basically designed to test this idea. We based it on a human variant called G359A. It's also dominant negative. It resides in the same DNM1 domain. It's present in at least 2 kids, probably more by now. But we expected the mice to be severely impaired, so we made a conditional model whereby when we add a recombinase that removes a stop cassette that prevents the expression of the exon showing the mutation, it removes that, the bad form of the RNA can then get active in any cell for which we have a driver CRI recombinase gene. And so we make the mice, they're born normal. And I should say that, well, they're actually born hemizygous. They have one missing copy of the RNA and one good copy. In the case of DNM1, those mice are completely normal, as are the people. So in the case of DNM1, we could do it like this. There are other ways of making such mice. And so we did need to choose what cells we wanted to test it in. And so to make a long story short, we wanted to make sure to have a robust challenge for the therapy. And so we crossed the conditional mice to a number of different CRI driver lines. And we chose a line that expressed the construct in all GABAergic, all inhibitory neurons. And so this plot here shows on the vertical axis the body weight, and this shows the first 4 weeks, 28 days of age. And you can see the growth curves of the control mice here are normal. And several of the growth curves of these CRI driver mutant lines are stunted, including this one here, the black one, where not only is there very delayed growth, but you can see the lifespan ends by the 3rd week, which is because they all die of a lethal seizure. And we thought, well, if we really want to see if this works, let's test it in those mice. And that's what we did. And so the experimental design is to inject the mice in the ventricle of the brain, newborn mice on the first day. And over the course of the next 4 weeks, monitor growth by measuring body weight, and then collect various molecular and cellular assessments along the way, and also monitor survival from those lethal seizures. And later, we'll look at other aspects of the mice. And so this is a complicated but catch-all slide that shows why I think this is very exciting. And I'll try to walk you through it easily. So again, it's the same kind of plot as before. The growth curve is shown on that axis, the 4 weeks of age, that axis. And you can see with no treatment or saline injection, the dotted line are the control mice. That's the normal growth curve, the normal lifespan, at least through 4 weeks. The mutant mice are these dark lines, and all but one of them died before the 3rd week of age. One managed to escape. They all had somewhat stunted growth. We actually made a version of the vector that lacked the cDNA and also lacked the knockdown. So we have the microRNA only and the cDNA only. And those look very much like the untreated or saline-treated stunted growth, truncated lifespan. But what's really cool is what's happened with the bivalent vector when you have both of them in there. And these green ones are what we're going to be focusing on. What is interesting that at the high dose we chose, this is as much we can get in to the ventricle safely. More than half of the heterozygotes died. The rest of them lived, but almost a third of the wild type, the control mice died. And so what we think is happening here is that Dynamin-1 knockout mice are very severely impaired. When you lack all Dynamin-1, they die by the first week of age. And so basically, we think this isn't toxicity, not autoimmune response. This is just plainly, we've turned these more and more like a complete knockout of the NM1 mouse. So we went to a dose half that and a dose a tenth of that. And together, these look really good. So only 1 in 25 mice died in the first 4 weeks. The hets of the mutants, and none of the control mice died at that time. The growth was almost completely restored at this lower dose. And so this is really, again, I think, incredibly encouraging for this kind of approach. The knockdown plus replace was sufficient and necessary for this. Neither alone would do it. And we can get the mice, most of them living and rectified growth. So we continued to watch those mice to see what was happening with them. We focused on that lowest dose. And as for the lifespan, we would say it's quite good. We have to utilize our mice to use for other experiments. So we haven't let them all go yet. It hasn't been this long since we started anyway, to the full 1.5, 2 year lifespan of laboratory mice. But of the 3 mice we let live that long, 3 lived at least 90 days. 4 other mice lived at least 60 days. 4 others 90 days. And 4 at least 6 months. They're beyond this now, so we have every reason to think their lifespan is approaching normal. We had a look in the females at reproductive behaviors. We mated 4 of these treated females to wild-type male mice. And the nice thing is they all had multiple litters, although only 2 of them raised their pups. And this was pretty unusual for the strain that we used here. And so this suggests that the females, while I would say cured of this severe disease, aren't completely normal. We've also noticed that some of the treated males are aggressive towards male siblings. Again, this isn't what we would expect for this strain background. So I would say the treated mice do very well, but they're not completely normal. I don't really know what we might have expected. A few other brief things. So we looked at the degree of transduction in the brain from this treatment. To make a long story short, at this dose, we really saw only modest transduction of all the neurons we could measure easily. This is 40% of those GABAergic neurons. By this transduction, we see only 10 or 12% of them present as co-staining for a tag we put on the cDNA and that cell type. So that was interesting. Am I missing a slide? I'm skipping a slide. Yeah. Oh, I should have had this slide before. I'm sorry. Anyway, my collaborator at Virginia, Matt Weston, also took cells out into primary neuron culture. And I don't have time to go through this slide, but basically he showed that those mutant mice have impaired neurotransmission, synaptic transmission from inhibitory neurons onto excitatory neurons there. And that's rescued to wild type levels by the viral treatment. And there are some other features that are also rescued. So this was nice to see some rescue at the cellular level and at the physiological level. Okay, so we then finally we use RNA sequencing for a couple of reasons. The first thing was we wanted to look at what was the level of the endogenous DNM1 level, DNM1 RNA that we knocked down, and how much of the viral RNA we were getting in compared to what we would expect to see from the endogenous. And so this was a little surprising at first. So we measure this at 2 weeks because we want to have the untreated mutant mice still around. And we saw across the cortex and the hippocampus a 22% decrease of endogenous DNM1. That seemed low, but if you calculate based on the amount of transduction on a per cell basis, we reckon this is close to 100% reduction of DNM1. So that seems fine. The level of replacement was also a little surprising. Less than 5% of equivalent of the endogenous DNM1 was the viral RNA was less than 5% of the endogenous. That also seemed quite low, but again, you do that back calculation about the percent of cells transduced, it comes up to about a third the level of these control mice. Also, it's known that the production increases up until about 3 or 4 weeks. So we expect it to be higher. And we've not yet assessed the codon optimization feature, the protein analysis coming. So these last few slides, the take-home messages, that degree of rescue I showed you required only modest transduction and modest RNA replacement. So we thought that was nice. So the last bit of data I want to show you is what happens to all the other genes when you put AAV in their brain and knock down DNM1 and replace it with artificially engineered DNM1. And so the goal here is to determine whether there were significant gene expression differences, increased or decreased, between mutant and control, and ask if there were any, were they changed after treatment? And so using kind of a standard for gene expression, there are two basic numbers here. We looked at treated and untreated control mice, and the number of significant gene changes here, expression changes, is really quite small, about 20 to 30, either lower or higher, when you compare that to the effect of the genotype, where you see about 500 significant expression changes going one way and the other in these untreated control mice. So this says to us that the treatment itself isn't, at least at 2 weeks, isn't really impacting the transcriptome that significantly compared to what's wrong in the brain. And so our questions going forward were, what biology are these genes involved with? What pathways, et cetera, et cetera? What went wrong? What's going wrong in these brains across the transcriptome? And does the treatment change that? And the last 2 slides are here. And so basically, to do this, we use something called functional annotation clustering, whereby the function of proteins encoded by genes is known to be in certain pathways and functions, biological processes, and they can be clustered into these categories. And so for the genes that were expressed lower in the mutant mice, you can see there was highly significant clustering, 7 orders of magnitude, when you compare the mutant versus the untreated control mice. And so it tends to taper off. And those categories were in things you'd expect: synaptic signaling, chemical transmission, transsynaptic signaling, et cetera, et cetera. Importantly, when you look at treated versus untreated control mice, there's no clustering. Even treated versus untreated mutant mice, there's not a lot of clustering. So this basically shows that all those problems that those mutant mice had were largely mitigated by the treatment. All these categories of genes that were caused by that mutation to be underexpressed is mostly rectified. And we see a similar thing, actually higher order of magnitude, with the location when we categorize those genes by their location in the places that we think we would have cared about: synaptic proteins, cell junction proteins, glutamatergic synapses, postsynaptic, dendrite, et cetera, et cetera. And the last interesting bit here is when we looked at the genes whose expression was higher in the mutant, there was also this clustering, but it was completely different categories of genes, and the degree of clustering wasn't so significant, although it's significant compared to these baseline comparisons. And the categories were things like, they were like a compensation category, so response to hormone, response to cell death, apoptosis, response to chemical homeostasis. So, so the genes that were lower are involved in the known pathways and genes like DNM1 and these diseases. The genes that were upregulated and were also rectified by treatment are seem to be in response type pathways. We did notice something unusual in the treated versus untreated mutant mice, as if there was something emerging, some genes in some of these categories as a result of treatment only in the mutants. And we're not quite sure what to make of that. I'd point out that, when you knock down all DNM1 and replace it with DNM1 that is expressed off of a ubiquitous promoter, somatically and mosaically, you're not exactly making a mouse that would normally be born that way. And so we are making some changes in these mice, but again, they don't have those severe disease features. So I'll summarize. I hope I didn't go through that too fast, but I'll summarize by concluding that this knockdown and replace therapy, not either, with this modest single dose. Sorry, my slides are being spastic. Mitigates the most severe disease features in these mutant mice. It also rectifies abnormal gene expression in relevant categories. And the effect of treatment alone was really pretty small. Treated mice look really good, but they're not completely normal, as I've described. There are some other changes, and for the degree they're not normal and those molecular changes, we can't really say whether those are emergent properties of the treatment or whether those are aspects of the disease that were residual that weren't rescued. We just don't know that yet. You have to do very directed experiments to get at that. I want to emphasize this was a very basic implementation of the knockdown replace approach. We used off a single transcript, a single genome, ubiquitous promoters. And so there's certainly ways to tweak it. And last, this knockdown replace part of it works best when the RNA is small enough for the vector. GRIN2 RNAs are a little too large. Amy talked about other delivery systems. There may also be other workarounds. It's possible you can cram GRIN2 genes in anyway. I know of another project where somebody has done this successfully, lower viral titers, but it works. And there's other approaches to this. So I'll stop. And I just want to acknowledge the people in my lab that did the work, and in Tristan's lab, particularly Divya, a talented senior technician, my colleagues at Nationwide Children's, Scott and Noah, and then Matt Weston. So thank you for your time, and you may have time for one question between us. Thank you.
Thank you, Dr. Frankel. I don't see any questions in the chat. Does anybody have any questions for Dr. Ramsey or Dr. Frankel? I don't think Dr. Ramsey's, you'll be on the, I don't think you'll be at the Ask Me Anything. So now's your time. We have just a minute left. Otherwise, we will go ahead and close the session. We have a 5-minute break, and then we have our breakout sessions 3 coming up. Thank you, Dr. Frankel, and thank you, Dr. Ramsey.
Thank you. Great work, Wayne.
Thank you. Same here. Same to you.
Take care.